Okay. Hello, everyone. This is a poster session here that we're hosting here at P&I's uh, summit. We have Valentina Francia, is that correct?
Yes.
Here from UBC to present her poster. I'm gonna, without further ado, I'll let her introduce herself further, and she can share her slides for her poster.
Thank you very much. And I want to thank you for being here, first of all, and then I want to thank you, Precision Nanosystem, for giving me the chance to present my research on the corona of lipid nanoparticles for gene therapy. My name is Valentina Francia, and I'm a postdoctoral fellow at the University of British Columbia in Canada and at the University Medical Center Utrecht in the Netherlands. So my research is focused on studying the corona of LNPs. And I know that I don't have to introduce LNPs in this context. So I will just say that I use as a model nanoparticle Onpattro, which has a composition as follows. So we have a ionizable cationic lipid, a phospholipid, cholesterol, and a PEG lipid, which are able to encapsulate siRNA. And LNPs, such and other nanomedicines, before reaching their targets, for example, here the liver, they enter in contact with biological fluids. When intravenously injected, the biological fluid is blood, or when inhaled, we have lung surfactants, or when locally injected, we have, for example, extracellular matrix. That interact with the surface of these nanoparticles, and these biomolecules are able to absorb on the surface of LNPs, forming the biomolecular corona. And the biomolecular corona is able to change the nanoparticle surface properties such as charge, size, hydrophobicity, and therefore it can influence the interactions between nanoparticles and cells. In particular, specific corona components can be recognized by specific cell receptors, such as in the case of Onpattro, in which ApoE is recognized by cell receptor on the surface of hepatocytes, and this recognition is able to trigger the internalization of Onpattro and the release of the siRNA. Therefore, corona is able to influence the nanoparticle targeting abilities of these LNPs and other nanomedicines. And the aim of my research is to study these interactions and how the corona influence the targeting in order to guide the design of targeted LNPs. First, in order to determine this correlation between the corona and targeting, we need to determine the corona composition. And to do so, LNPs need to be incubated in relevant biological media, and afterwards, these corona nanoparticle complexes has to be isolated and characterized by different analytical techniques, such as, for example, SDS-PAGE or mass spectrometry. And then this LNP can be tested for cellular uptake and silencing efficacy as well. The first step, the incubation step, might look trivial, but it hinders several challenges. The first challenge is that we cannot totally reproduce an in vivo environment in vitro. Incubation parameters such as the choice of dispersion type, the species, the disease type, or the dispersion concentration, dispersion temperature, and the incubation time, they all can influence the formation of the corona, and therefore they influence the way cells recognize these objects. Said so, any change in these parameters can change drastically corona composition and therefore the LNP's targeting ability. Therefore, controlling these parameters is fundamental for making in vitro studies, for making in vitro studies that are predictable of the in vivo situation. So after incubation, LNP's corona complexes needs to be isolated from the dispersion components. In this case, we are talking about blood. And this isolation must, at the same time, preserve the corona and particle integrity and remove all these unbound proteins and lipoproteins present in the biological fluids. There are several techniques that can accomplish that, and the choice of your technique depends on the material that you have. In the case of LNPs, this is particularly challenging since LNPs have the same size and density of natural nanoparticles, such as HDL, LDL, chylomicrons. And therefore, all those techniques that are using size or density to separate particles are not suitable for LNPs. One example is size exclusion chromatography, which basically consists of a gravity column, which is filled with porous resin that is composed by different materials. In this case, for example, the most used for corona study is saprotropin. So the sample is injected on top and then is separated by size, and different fractions with different sizes are collected. However, the same fraction that contains LNPs might also contain those natural nanoparticles which has the same size as LNPs, and therefore this method is not ideal if we want to characterize precisely the composition of a particular LNP. However, it can still be used if you want to compare different samples among them, and then taking into account these differences in this kind of background noise that is generated by the presence of these natural lipoproteins present in biological fluids. Therefore, in our lab, we decided to use another technique, which is magnetic separation, which is able to retain in a magnetic column magnetic nanoparticles. However, to do so, we first need to have these magnetic LNPs, and therefore we formulated LNPs that are magnetic based on the composition of Onpattro, as listed here, and with increasing amounts of 5 nanometer iron oxide magnetic nanoparticles in their core. As you can see here, at increasing concentration of iron oxide during the formulation process, we have an increase in incorporation efficacy of iron oxide. However, we still have differences in size between LNPs, and also differences in incorporation of these iron oxide particles. There are LNPs that are completely empty, and other LNPs that instead have up to 50 iron oxide per particle. Therefore, we decided to run this sample through a magnetic column and see if our magnetic column could retain these iron oxide particle, and if we could get rid of the empty LNPs. So the samples were loaded in a magnetic column in the presence of a magnet. The flow through and washed were washed with PBS, and therefore theoretically all the iron particles are retained in this magnet, whereas all the particles without iron should be washed away. Then the magnet is removed, and then the sample is eluted, and this is the elution fraction. And as you can see here, the particles recovered are filled with iron oxide particle. Here on top, you can see the lipid composition of the lipid concentration of the different fraction in the 4 different formulations. And as you can see in the first formulation, most of our sample is lost in the flow through and in the wash fraction due to the fact that these particles are not magnetic enough, or they don't contain enough magnetic iron oxide in their core. Whereas in the formulation B, we managed to recover up to 30% of particles. And therefore, we decided to proceed with this formulation for our studies. In particular, it's very interesting to notice that in the wash fraction, the incorporation is very low. So that most of the particles that are washed away have just a low number of iron oxide per particle, compared to the elution fraction. And at the same time, we can see that in this fraction, in the wash fraction, the average particle size, the LNP size, is lower compared to the particle size present in the E fraction. And this is because there is a correlation between LMP diameter and the number of iron oxide incorporated in each LMP. This first magnetic separation allows us, therefore, to recover a kind of homogeneous formulation, which is composed by slightly bigger particles, which incorporate up to 50 iron oxide per LMP in their core. So these magnetic particles were incubated, in this case in human serum, and after this incubation, the corona nanoparticle complexes were run again through a magnetic column. And theoretically, all the proteins and lipoproteins which are not magnetic should be washed away, whereas the nanoparticles with their corona should be retained in the column and then eluted here. And this is exactly what happens. And it's even more obvious when this technique is compared to size exclusion chromatography. So basically the same sample was divided in two parts, and each part was run through size exclusion chromatography or through a magnetic column. And here you can see that the lipid stocks, so the amount of lipids that are injected into each column is exactly the same. However, the amount of lipid that are recovered in the case of size exclusion chromatography is 3 times higher compared to the lipid that are injected. And the reason is that in the fraction recovered by size exclusion chromatography, we also have a lot of these vesicles still present, which increase, of course, our lipid recovery. Instead, in the case of magnetic separation, we are able to recover only 1/3 of our sample, but here we are sure that the sample recovered contain only, or at least for the most part, the corona of LMPs. It's even more obvious when we look at the protein recovered. So when the sample have the same amount of lipid recovered, measured by fluorescence, you can see that in the case of size exclusion chromatography, we recover 3 times more proteins compared to magnetic separation. And this means that when we analyze this sample by mass spectrometry, for example, the amount of protein that we get is 3 times higher than the one that should be. So 2/3 of these proteins are probably coming from all these protein and lipoproteins present in the serum, and therefore they will interfere with our analysis. So once these corona nanoparticle complexes are isolated, then they can be assessed by gel electrophoresis or mass spectrometry. And here you can see, for example, that the same sample incubated in human serum of mouse serum is run through this polyacrylamide gel and then stained by Comassi blue. And here you can clearly see the protein profile of the 2 samples, and you can see that there are completely different bands. On the right instead, you can see the list of the top 10 most abundant proteins in the corona of these LMPs that can be classified based on their abundance, in fact. So to conclude, we saw that the corona can influence the LMP targeting capabilities, and that in vitro study must take into account the biological interactions in order to be predictive of the in vivo situation. We saw that the incubation step is critical in order to form a biologically relevant corona. And we also saw that many isolation techniques are available. However, not all of them are suitable for every application. And to conclude, the next step will be determining whether the composition can help us predict targeting in a personalized way. So can these differences in the corona composition that we can see in different samples help us predicting how to design better nanomedicine? And with this, I want to thank you for your attention. I want to thank you, Precision Nanosystem, and I want to thank you, to thank my colleagues and supervisor.